Okay, welcome guys. So what I'm going to do today is just give you a very brief explanation of the whole 5' to 3' direction that we see on DNA. So when we're talking about transcription and translation, it's very easy to see that we have enzymes that are being read in this 5' to 3' direction. But if we're unsure of what 5' to 3' means, we may come into some difficulty. So here on the slide, we can see that we've got phosphate groups attached to, we can see that we've got like phosphate groups. If we just do like a red, actually, we'll have to do a black. So we have this phosphate group over here and here, etc., etc. Okay, continuing on and on. Now, we can see that this phosphate group is added to a carbon, as is this phosphate group is added to a carbon, and then carbons and carbons. The numbers, 5' to 3', etc., depend on the number of carbons on the sugar. So if you look at these numbers here, I know they're quite small. Okay, you can see just 5' 4 there. So these numbers are effectively the carbon number. We start the first carbon is that the first carbon is the carbon that's bonded to the base, for example. So in this case, it's thymine. So this thymine is bonded to the first carbon in the sugar. It's important to think of the sugar like a central atom. Okay, so we have the sugar over here, and off of that sugar we have the base, and then off of that we have the phosphate group, which I'll just draw as a P. Okay, so you may have seen this diagram before. So the sugar is really acting, in this case, as the central. Okay, this is the central compound. It really is the central compound where the base and the phosphate link on. Okay, so you can see here we have this central sugar running down the middle of the phosphate. The phosphate has a shoot off there, and then the base shoots off left to right. So it's really the sugar that we're counting when we're talking about 5' and 3'. So let's just have a bit more of a deeper look into the 5', 3', and why is it 5 and 3 and not 1 and 2? What do we really mean when we're talking about this? Well, as if we just get rid of these things, well, we can see then that if we have our second carbon, we have our first carbon, our second carbon, and then on the third carbon, if we just look at this little diagram here, you can see that there is a carbon. There is a carbon here, it's just not shown in this line diagram. Then we have a phosphate group being linked. So there's a phosphate group linked to this carbon, and this carbon is the third carbon, and we'd call this section 3'. This is the third carbon on this sugar, sugar here. The exact same thing could be said if we count 1, 2, 3, 4, 5. This little, what almost looks like a methyl group. We have this sort of methyl group up here. This is the 5th carbon, and then here we also get this phosphate group. Okay, so we'd call this section 5'. This is basically how DNA is read. DNA is read on a 5 to 3' orientation. So if we are looking at a specific enzyme, say we have an enzyme that's called DNA polymerase 3. DNA polymerase 3 will effectively link onto this section. Okay, if we were sort of just colouring it in, basically this is DNA polymerase 3. DNA polymerase 3 will attach itself to the phosphate backbone, and it will move from 5 to 3'. So it reads the phosphate groups and sugars in this direction. This is how we get DNA being read and transcribed, even transcribed and everything. Enzymes tend to favour this 5 to 3' direction. Okay, so hopefully that summarises quite nicely what I mean by 5', what I mean by 3' direction. And then if we were looking at just one more thing before I let you guys go, I know this is recorded, but still, if we have a look then at what this means for DNA replication, because this is a topic that may be requiring a bit more explanation, we can see then that if we have two DNA bases, two DNA strands, okay, one that starts off 5' to 3', and the other is obviously the opposite because they are complementary to one another. 5' to 3', 3' to 5'. If we have an enzyme, or we'd have two enzymes technically, okay, if we have two enzymes on here, okay, say this is a DNA polymerase, let's just draw this a bit better. Okay, let's just say this is DNA polymerase and this is DNA polymerase. These are on different strands of the template. They will move in different directions. Okay, what we would see is we'd see this DNA polymerase moving this direction from 5 to 3, and this DNA polymerase moving this direction from 5 to 3. Okay, this is how we understand leading and lagging strands as well, based on which way the replication fork is. So if we say that the replication fork is over this side, let's just say this is the fork over here, then we can see that this strand would be termed the leading strand. Okay, this strand over here that's moving 5' to 3', yeah, it would be moving into the fork. Okay, so this one would lead, this one would lead, and this one would lag over here because it's moving away. Right, I hope that has been a good little summary of how DNA replication really takes place using the 5' and 3' description on the phosphate sugar backbone of DNA. Please utilize the other lectures and other resources on Moodle, and this should really help you guys in your exam preparations. Thank you.